I'm Koppers. I'm a professor here in the College of Earth, Ocean and Atmospheric Sciences and a director of the Argon Geochronology Lab. This is a lab that allows us to date materials from the Earth that we collect from the ocean floors, the continents or even from the moon. And when we get those materials, we process them here in the lab to make them ready for the analysis. The analysis happens right over here in my lab. So in this lab we have two instruments that are basically behind closed doors because we use CO2 lasers and we use radioactive materials. But the cool thing is that when we have prepared the rocks in our other labs, we actually, we take only a very minute amount. So we collect maybe kilos of samples in the field, where we all downsize it and clean it up to maybe only 5 or 10 milligrams of material. That material we load up in our laser system, which is right here. I will walk around a little bit. So right here we have what we call a laser chamber, and in there's a copper tray that actually has those 5 to 10 milligrams of materials that we separated in our other labs. What is important is that those samples are, we need to get the argon out of it. Unfortunately, there's also a lot of argon in the atmosphere. 1% of the air that you breathe is argon. So when we do our analysis, of course, we cannot have it contaminated with all the argon in the air. So when we do our analysis, it's done under an ultra-high vacuum. So the noises you probably hear now in the background are all the pumps pumping down our instrument to a really, really good vacuum. But if there's a really good vacuum and that sample in that tray, so we heat it up with the laser, which is this piece of equipment. Basically the laser goes from this gray box down into the sample chamber. When it hits the rock, it actually starts heating up that material. And that 5 to 10 milligrams of material will basically release the argon gas. It gets collected in the sample chamber, and as the gas, it starts expanding. And it expands all the way through this line. There's a small little pipe here, and then the gas travels through that on the inside and gets into this next section. In the next section we clean it up, because the laser not only releases the argon, it also releases CO2, methane and other gases we are not interested in. Once that's cleaned, maybe after 90 seconds or 10 minutes in some cases, then we are ready and we actually let the gas expand into this mass spectrometer. There we can separate out the isotopes, measure the ratio between the isotopes, and that gives us the laser. Right. As I said, we are working with radioactive materials, right? So we need to have a special room, which is right here, where we actually can store and handle the isotopes in a very safe manner. So that's one of our other labs that we have. Okay, so this is our mineral separation lab. This is where we get those samples from the field, right? Kilos of materials from the ocean floor that we need to downsize to the 5 to 10 milligrams. Now, one of the ways that we do that is with magnets. We have here what's called a France magnet. And actually when you pour a sample in it that has been ground up to basically sand particles, you can put it on here, and it will basically vibrate down this little slide, and the magnet actually pulls out grains that are a little bit more magnetic than the other. So we want to get CPX out, or we want to get plagioclase out, which are two minerals. Different strengths of the magnet allow us to do that, and that's how we purify our samples. And we want to get as pure as we can go. We also often get samples from the ocean floor or anywhere around the world. Nature has been working on it, like in the oceans, the seawater penetrates the rocks and actually can alter or weather it. And we need to get rid of that alteration, because that actually is influencing the analysis that we want to get. And so what we often do is with those samples, we clean them with acids. We use hydrochloric acid and nitric acid or hydrofluoric acid. Basically it's to get the bad stuff out of the rock. We basically dissolve alteration minerals, clay minerals, zeolite, things that we're not interested in. And that's happening right here. So this is basically the lab where we do the final stages of the cleaning of the rock. So when everything is processed, everything is cleaned with the acids, separated with the magnets, we come here with the final pieces of the sample, and we use microscopes to basically go over grain by grain and basically pick out the ones that are bad. So the bad ones we throw out, the good ones we keep. And this is a very labor intensive work. It can take from maybe an hour per sample to sometimes days of picking behind the microscope. So our lab is basically famous around the world for anything that we do in the oceans. So any volcanic rock that is formed there, whether it's the oceanic crust, volcanic islands, seamounts, large igneous provinces, we can date them, and people come to us to get their rocks dated.